Hello dear viewers, myself Nasir and you are watching Mr.Zoologist YouTube channel. Today we are going to discuss about indices of toxicology. In actual, the concentration of toxic agents, which is most important factor in evaluation of its toxicity, is a part of indices. Small amount of some chemicals may cause intense toxic effects, whereas others large quantity may be needed to affect a biological system adversely. Even most safe chemicals may be harmful if high doses are administered or given. Toxic effects of substances are evaluated by concentration at which deleterious responses appears. These responses can be toxic dependent upon the concentration provided. If we talk about common indices, there are four types of indices used commonly. These include threshold dose response curve, sublethal dose response curve, toxic lethal dose response curve, and therapeutic dose response curve. First of all, we are going to look at threshold dose response curve. Minimum or smallest dose of a substance which causes adverse effect or change in organisms as determined by the most sensitive biochemical and physiological methods, even if no external indication of such a change is present. Means to say, some chemicals are so dangerous that they might not show symptoms in the form of external symptoms, but they internally damage the organs or the body in a adverse way. And if we talk about threshold dose, it is a chemical agent is the dose at which its toxic action just starts. Minimum to minimum dose which can provoke a negative response in an individual is called as threshold dose response. And threshold limit is marked by threshold concentration limit. That concentration limit may vary from individual to individual because toxicity of any substance depends upon the gender, age, and working of an individual. And that is a threshold curve. You're looking in the current slide. On the x-axis, we have taken dose in mg per kilogram body weight of an individual, whereas on y-axis we have taken response. As initially we have given the dose in initial administration, there was no significant effects or there was no response observed. But as the time progresses and the dose concentration increases with relative to the body, the curve also goes higher and higher in the form of response as the concentration of the toxicant is increasing. That shows with the passage of time as dose amount increases as well as its response also increases. And in the second, we are going to look at sublethal dose response curve, the amount of chemical agent required to cause appearance of some observable symptoms or visible manifestation of toxicity without resulting into death of test organism. Means to say, there will be toxicity of the chemical, but that will not lead to the extinction or death of the organism. Any dose of toxic agent between threshold dose and the lethal dose, which usually manifests itself in form of certain responses, the organism undergo observation exhibits, but death does not occur. As I have told you earlier that there is no relation of death with respect to sublethal dose response. The response of sublethal dose can be used to determine effective dose, which is another index of toxicity denoted by environmental development toxicology. And that is a sublethal curve. As we have increased the dose, and that if we talk about the LD50 concentration, that is used for a population in which if we administered a certain chemical toxin, that chemical is a toxin, and if that toxin induces 50% mortality of the whole population being under examination of these toxins. And third one is the toxic lethal dose response curve, dose of chemical agent which causes an individual to be killed. Here the death will occur probably. Toxicity of the most of the chemical agent varies from individual to individual, and factors like strain, age, weight, food type, caging, volume, and type of suspension medium, method of administration, all these affect the toxicity of a substance to the individual. It is not possible to estimate toxical lethal dose accurately on an individual. It is usually assessed on groups of individuals. We will never carry out a experiment on single individual. Any experiment that is conducted to test toxicity consists of control groups as well as experimental groups. The data is converted to probit units to straighten the regression line. That regression line tells about the marked deviation from the center line for the control group as well as for the experimental group. And if you talk about it is in form of regression line, that mortality or mortality in probit units at any desired dose is determined. That is a toxic lethal curve that is showing the mortality of the individuals in the population that have been administered that toxin. And fourth one, the therapeutic dose response curve. Chemical agents used in practice therapeutic medicine up to a certain concentration produce desirable responses. These does response or dose response curve plotted for appearance of any desirable symptom is called therapeutic. Means therapeutic is an actual treatment dose response, which is similar to toxic dose response curve that tells about how much been done in the individual. That is a graph or curve showing the therapeutic curve. On x-axis we have taken the dose, and on the y-axis we have measured the response. And that slope shows maximal efficacy as the potential of the medicine increases. So the amount of dose decreases, as well as individuals become to recover from that toxin's toxicity. So in if we talk about uses of term concentration, used in case of aquatic organism usually instead of dose. Concentration is usually used in aquatic environment, and dose is used in mostly terrestrial environment, because the amount absorbed by organisms can be determined. Dose or amount of toxic agent absorbed varies according to a number of factors. The concentration of agents in air or water can be determined accurately, but that depends upon the concentration units being used